Hello, class. Today we're going to lecture on the other group of seed plants, which is the angiosperms. Our outline is as follows. First, we'll learn about the phylum to which the angiosperms belong, which is Magnoliophyta, focusing on gametophyte development. Then we'll learn about pollination and fertilization and the resulting seeds that develop inside of fruits. We'll talk about some specialization trends in the flowering plants. We'll say a few words about pollination ecology, which is to say what are the features of the flowers and the pollinators that visit them. Then we'll end by talking about herbaria and preserved plants, as well as a review of the dicots versus the monocots. Angiosperms are plants with seeds contained within a structure. Recall that the root word angio means enclosed, so these seeds are enclosed inside of the carpel. In review, the carpel is made up of a stigma, a style, and an ovary, and the ovules are found inside of our ovary. For the angiosperms, flowers and fruits are the most distinctive part of this group. In fact, it's because of these flowers and fruits that this is the most diverse and largest in terms of number of species of any other phylum in the plant kingdom. So what are the shared features of the angiosperms on the tree of life? Well, as we previously said, the ovules are found inside of a carpel, and there's also a stigma present because there has to be somewhere where those pollen grains may land and be received in order for fertilization to even be possible. Stamens are typically with 2 pairs of pollen sacs, and double fertilization is common in the angiosperms. The double refers to the fact that there are 2 sperm nuclei that are needed in order for an embryo to be produced. Lastly, the phloem tissue is composed of sieve tube members and companion cells. Angiosperms are hugely diverse. They are one of the major groups of the seed plants that are currently found on our planet today, and they have over 260,000 living species that are found in over 450 different families. They occupy every habitat on Earth except for extreme environments such as the highest mountaintops or regions where there's just too much ice. There are many different strategies that they use in order to grow. Some of them, for example, grow as epiphytes, meaning they grow on other plants. Others are parasites. We find herbs, woody plants, aquatic plants, vines. So many growth strategies are possible. Angiosperms have been divided into 2 large classes. We can break down this phylum Magnoliophyta into the Magnoliopsida and the Liliopsida. The Magnoliopsida is the dicots, and we can see the spelling of that word on our screen. DNA and other evidence suggests that the 2 groups of dicots are recognized as 2 distinct categories. The so-called eudicots, and notice the spelling on that word, it begins with the letters EU, and the magnoliids. And these magnoliids are the more primitive of the dicot groups. Then the Liliopsida is the monocots as we know them. Now, in addition, on the tree of life, you'll see some other families and groups appear, including the most primitive of all, which is the genus Ambarella. And again, you can see the spelling of that word on the screen. They have flowers that are male and female, and they are extremely primitive in form. They are endemic to an island chain called New Caledonia, which is found off the coast of Australia, and it's actually home of thousands of super rare plants that are found nowhere else on our planet. Angiosperms reproduce sexually. There are sperm and egg cells involved. They are heterosporous, meaning that they produce both a mega and a microsporangium. And just like the gymnosperms and the seedless vascular plants, they are sporophyte dominant. The sporophyte generation for their alternation of generations life cycle makes up most of the plant. The female gametophytes are wholly enclosed within the sporophyte tissue and reduced to only a few cells. Again, this is that evolutionary move towards containment and a reduced gametophyte. At maturity, the male gametophytes consist of a germinated pollen grain with 3 nuclei. And this is going to be important because it's going to play a role in that double fertilization that we talked about previously. The female gametophyte in angiosperms develops as follows. First, a diploid megasporocyte, which is another word for the megaspore mother cell, differentiates from all other cells in the ovule. This megasporocyte undergoes meiosis and produces 4 haploid megaspores. 3 of these megaspores break down and degenerate, and 1 of them survives. The integuments that surround this structure differentiate and eventually become the seed coat. Third, the female gametophyte, which is referred to as the megagametophyte, is formed, and it consists of a large sac that usually contains 8 nuclei in 7 cells. And for the purposes of our exam, I want you to be able to know the definitions of the polar nuclei, the synergids, and the egg cell. And these are going to play a role in double fertilization, which is what we're going to discuss on the next slide. Here we can see a diagram that shows us the relationship of the female gametophyte as it develops. And remember that gametophyte is developing from the megaspore that survived. Now, in the male gametophyte, that is going to develop in the anthers. And this is happening at the exact same time as the female gametophyte is developing. Now, the name for the male gametophyte is the microgametophyte. Remember, mega is the root word associated with female parts, and micro is the root word associated with male parts. The microgametophyte develops from 4 patches of tissue that differentiate from the main cell mass. This contains diploid microsporocyte cells, which undergo meiosis and produce a quartet of microspores. In this diagram, it shows us a lily anther in cross-section in which the pollen grains are forming. The microspores are going to give rise to microgametophytes, and the microgametophytes are the pollen grains. After meiosis, the haploid microspores in the pollen sac undergo several changes. For one, the nucleus in each microspore divides by mitosis. Next, a 2-layered wall develops around each microspore. These microspores are divided into pollen grains with a tiny 2-celled microgametophyte contained within. Now, it is compatibility chemicals in the wall that are responsible for stigma recognition of this pollen grain. Within this pollen grain, we find a tube cell which grows the actual pollen tube, and we have a generative cell which divides to form two sperm. So next up, we now understand how the mega and micro gametophyte are developing and play a role in forming our gametes. So now we have to transfer the sperm, which is found in the pollen, to the stigma so that it may develop and contribute those sperm nuclei to the egg. Now, pollination is simply the transfer of pollen grains from an anther to a stigma, and fertilization is where the sperm and egg actually fuse. Now, pollination happens every time a pollen grain lands, but the pollen grain will not necessarily be received. Fertilization, on the other hand, may not occur until days, weeks, or even months after pollination has taken place. After pollination, further development of the male gametophyte may not take place unless the pollen grain matches the following criteria. For one, the pollen grain should be from a different individual of the same species. That would be the definition of outcrossing. And two, the pollen grain should be from the same plant only under special conditions. This would be an example of self-pollination, which doesn't happen very often. However, there are some plant groups in which this is the primary way that they reproduce. As long as suitable conditions are met, a pollen tube will grow down between the cells of the stigma and style until it eventually reaches the ovule micropyle. Once the pollen tube reaches the micropyle, it continues to the female gametophyte. And once it reaches this area, it discharges its contents. This is where double fertilization finally occurs. One sperm migrates to the egg and forms the zygote, and the other sperm unites with two polar cell nuclei, producing a triploid endosperm. Look at that root word tri. Tri means three. So therefore, triploid is referred to as 3N. And this is a very important embryo food source for the angiosperms. This shows us the generalized angiosperm life cycle, of which we should understand all of the steps that we have lectured on in the previous slides. All right, so now we're ready to move into our next stage of the lecture, which is about specialization trends. The fossil record suggests that flowering plants first appeared about 160 million years ago. So this is what we refer to as the late Jurassic period. Now, based on the fossil record, we believe that primitive flowers had numerous spirally arranged parts that were not fused together and were highly variable in number. So while we learn these rules of monocots are in parts of 3 and dicots have parts in 4 or 5, these primitive flowers have lots of different parts. The flowers were regular in shape and contained many stamens and many carpels. Examples of primitive groups of flowers include the magnolia and the water lily, which is in the genus Nymphaea. In advanced flowers, the receptacle is typically fused to the ovary. As a review, when the ovary is embedded in the receptacle, it is said to be which of the following, inferior or superior? What do you think? The best answer is inferior, because it is embedded and surrounded by the receptacle so that it appears as if it is below the other parts of our flower. On the other hand, when the ovary is produced on top of the receptacle, it is said to be superior. Now, more advanced flowers tended to become irregular in shape, which is to say that they had a bilateral symmetry. In general, the evolutionary trend is towards the reduction and fusion of flower parts because these flowers tend to be more specialized. And that's a bit counterintuitive because you might think that a flower with more parts is more advanced, but in actuality, we believe it's the opposite. Now remember, the term complete refers to the fact that a flower possesses all four whorls: calyx, corolla, stamens, and carpel. An incomplete flower is missing one or more of these whorls. We can further break down flowers into the groups perfect versus imperfect. Perfect flowers are bisexual, meaning they have both male and female reproductive parts. But imperfect flowers are unisexual, meaning they only have male or female parts in each single flower. We can further break down these angiosperms into the groups monoecious versus dioecious. And notice the spelling on the screen. Mono means one, and ecious means house. So monoecious angiosperms have both male and female unisexual flowers, versus dioecious angiosperms only have male or female unisexual flowers on a single plant. This is a poster that was actually made by one of my colleagues from graduate school. He studied salvia in Central America, and he put together a poster that shows all of this floral shape of this genus Salvia from the mint family, all of which is bilateral. Here's a picture of a California native tree called the tan oak, and they exhibit imperfect flowers and are said to be monoecious. Now we're going to learn about pollination ecology, and we're going to learn the characteristics of flowers and their pollinators. Overall, we believe that throughout the evolutionary history of the angiosperms, pollinators have co-evolved with plants over millions of years. The first pollinator is the bees. There are 20,000 species of bees around the world that pollinate flowers. Characteristics of bee-pollinated flowers include the fact that they are very colorful, they are fragrant, and they produce nectar with nectar guides. And these nectar guides are visible in ultraviolet light only, but it allows the bees to find those flowers and that nectar source kind of like a bullseye. Number 2 is moth and butterfly pollinated flowers. These flowers also have sweet fragrances and nectar. They have a shape that is conducive to the long tongue of these flying insects. Now, moths tend to come out at sundown and pollinate flowers right at dusk or throughout the night. So therefore, the flowers tend to be white or yellow to have that contrast against the darkening skies. Butterflies tend to go to brightly colored flowers, such as blues or yellows or oranges, but either way, the nectaries are found at the bottom of a corolla tube that is situated for their specialized tubular mouthpart called a proboscis. This is a picture of some moths that are pollinating some flowers in the desert that I observed over spring break. There we go. I pressed play on this video I took with my iPhone, and there we can see these moths are huge, the size of my fist. They're referred to as hummingbird moths or sphinx moths, pollinating these flowers at sundown. Our next group is the beetle-pollinated flowers. Beetles tend to visit flowers with a strong, yeasty, spicy, or fruity fragrance to them. However, the flowers are usually white or dull in color because beetles don't have keen visual senses. And they're also really bad at flying, so those beetles tend to be attracted to big open flowers that's easy for them to land on. Also, some of these beetle-pollinated flowers don't produce nectar at all. Instead, they make tons of pollen, and so the beetles will eat some of the pollen, but some of it gets stuck to their body, and so outcrossing happens. Fly-pollinated flowers tend to be unique, especially those that are pollinated by flies with short tongues. These are going to be flowers that usually smell like a corpse or rotten meat. They attract flies in because the flies think it's a dead body that they can lay their eggs in for their maggots to be born. But it's a trick because it's a flower, and the flies end up doing that outcrossing for these flowers. For example, on your screen, it's a picture of the fly-pollinated starfish flower from South Africa. It's very bizarre member of the milkweed family, and the large flower has the scent of rotting meat to attract blowflies and flesh flies from miles around. The flower actually gets hot like a dead body to trick those flies to enter. Now, not all flies have short tongues. Many of them have long tongues. And in fact, if you're out on a nature hike and you see some pollinators working on flowers, not all of the things that look like bees are actually bees. Some of them are long-tongued flies. Next up, we've got flowers that are visited by birds, and bird-pollinated flowers tend to be bright red or yellow, and they usually have little, if any, odor. And the reason for that is because birds can't smell. They don't have a very keen sense of smell. They have a great visual sense, though, so they're really going to be attracted to those bright red or bright yellow colors. Also, the shape of the flower needs to be such that it's going to facilitate the bird's beak as it's trying to get that nectar reward. Also, bird-pollinated flowers have to make tons of nectar because birds can be voracious drinkers of that nectar. And lastly, we've got our bat-pollinated flowers. These tend to be flowers that open at night because bats tend to be active only at night. An example is very famous from the southwest deserts of the United States, the iconic saguaro cactus with its huge flowers that open to allow for those bats to visit. And while we learn these trends, many flowers are actually pollinated by multiple animals. Birds, bats, and bees might all visit the same flower, even if we're talking about the saguaro cactus. And here is an example of monkey pollination. Actually, just kidding, that was a joke. So this is an orchid. It's actually in the genus Dracula, and this orchid flower looks just like a monkey face. It's so bizarre. The reason for this bizarre shape is to attract a pollinator, which is a type of bee that comes and visits this fragrant orchid that smells just like ripe oranges. All right, so in the last part of our lecture, we're going to talk about herbaria and plant preservation. So an herbarium is a library of dried and pressed plant specimens, but it's usually going to include other kingdoms as well. An herbarium typically has algae and or fungus, depending on the collection. Most scientific research facilities, including universities, have an herbarium. When I was in graduate school at San Francisco State University, I worked in the herbarium there as I was training to become a botanist. Now, these dried flower samples actually can last for hundreds of years. It's very important for conservation reasons that we keep track of these flowers on our planet. Also, when you're doing molecular research, you can actually sample from these herbarium sheets. You can still get DNA out of these dried leaves, even 100 years or more later after they have been collected. And a quick comparison of the dicots versus the monocots. Review: the flower parts of the dicots are typically in 4s or 5s. Their pollen is triaperture, meaning it has 3 pores or openings. It has 2 cotyledons, net-like veins on the leaves, primary vascular bundles in the stem arranged in a ring, and true secondary growth with a vascular cambium is present in all woody species. In the monocots, the flower parts are in 3s, typically. The pollen is monoaperture, meaning 1 opening. It has 1 cotyledon, parallel leaf veins. Primary vascular bundles in the stem are complexly arranged, and they do not have true secondary growth as we learned with the dicots. All right, that's our review of all things angiosperm. We've now learned about all of the anatomy and morphology of both the gametophyte and the flowers themselves. We've talked about pollination versus fertilization, specialization trends, pollination ecology, herbaria, and a quick review of the dicots versus the monocots. That's it for this lecture today. Please email me with any questions, and thank you very much for listening.